Hi, great to see you back. Don't forget to like, share and subscribe to my channel. We're looking at cell structure in topic 1 of cell biology. Today we're going to look at microscopy with some tips, linking words and exam questions. There are different sections of microscopy: evolution of a microscope, the formula for magnification, using the light microscope, and how to prepare a slide. And we will look at all of these in today's video. Let's first look at the evolution of a microscope. For the GCSE level, you must know about light microscope and an electron microscope. The light microscope uses light and lenses which forms an image. It magnifies things to make them look bigger, and it allows you to see individual cells and large subcellular structures such as the nucleus. On the other hand, the electron microscope uses electrons instead of light. It allows you to see things that a naked eye wouldn't be able to see. It does that by having a higher magnification and a higher resolution. Resolution is the ability to distinguish between 2 points. That means it gives you a sharper image. Electron microscopes allows you to see very small structures like chloroplast, mitochondria, ribosomes and plasmids. To do the calculations, you must know the formula for magnification, which is magnification equals image size over real size. Let's have a look at an example. In this example, the specimen is 30 micrometers wide. That means the real size. We must calculate the width of the image specimen, which means the image size. Under magnification of 100. To do this question, we have to rearrange the equation. An easy way to rearrange the equation is to use the triangle method. The tip for the triangle method for this formula is to remember miss with only one S. We have our M, which is our magnification, and we have our real size, which is 30. If we multiply these together, we get 3,000. But the answer is given in micrometers. So we have to convert the answer to millimeters, as it says in the question. To convert the micrometers to millimeters, you can divide the value by 1,000. So the value we have is 3,000. We divide that by 1,000, which gives us 3. So our answer for this question is 3 millimeters. In this course for the microscopy section, you must know how to use a light microscope and also how to prepare a slide. Before we know how to use the light microscope, we must be able to label it. Here we have a diagram of a microscope. This is the eyepiece. We have 2 adjusting wheels, a coarse adjusting wheel and a fine adjusting wheel. We have a light source, the stage and the objective lens. So how do you use a light microscope? First of all, number 1, you must clip the prepared slide onto the stage. Select the lowest objective lens. You use the coarse adjusting wheel to move the stage. You look through the eyepiece and use the coarse adjusting wheel to focus roughly. The fine focus wheel does the fine adjustments. You can change the magnification to see with greater magnification. If you do this, you must refocus. This brings us to the part of preparing an onion slide. To prepare an onion slide, you must add a drop of water in the middle of the slide. Take an onion and separate its layers. Using a tweezer, peel off the epidermal tissue, which is the thin layer of tissue, and place this epidermal tissue onto the slide with the water. You must add iodine solution and cover this with a cover slip. That must be done very carefully to avoid air bubbles. We must be able to recognise a diagram of an onion slide and be able to label it. Here we have the slide, we have the water that the epidermal tissue will be placed on. Place some iodine solution, which is shown as the brown colour, and we cover that with a cover slip. Tip to remember is to be able to do this carefully to avoid air bubbles. This brings us to the last section, exam questions. Diagram shows two cells, a bacterial cell and a plant cell. Both cells are drawn the same length, but the magnification of each cell is different. You can see the magnifications at the bottom. For the bacterial cell, the magnification is 50,000, and for the plant cell, the magnification is 500, as shown here. The question, it tells us that both cells are drawn at the same length, but the magnification of each cell is different. The real length of the bacterial cell is 2 micrometers. Calculate the real length of X of the plant cell. Give your answer in micrometers. So this is quite a tricky question. So first of all, if we recall our triangle, and in our triangle we remember the miss without the double S, we start our M on the left side and write that out. If we go back to the question, it tells us the real length of the bacterial cell is 2 micrometers. So first of all, we will work out what the image size for the bacterial cell will be. So the bacterial cell is 50,000, which is the magnification, and its real size is 2 micrometers. So we'll multiply by 2. So it's 50,000 times the 2, which will give us 100,000. Once we have that figure, we know for the bacterial cell, this is 100,000. We have to calculate the real length of X of the plant cell. Now we know what the image size is, we've just worked it out because it's telling us both cells are drawn at the same length, which is 100,000. Now we use the triangle method again. Now we have the image, which is 100,000, and we have the magnification, which is times 500. This time we have the image size and we have the magnification, so we will divide the 100,000 by the magnification, which is 500, which will give us the real size, which is what we want, the real length of X. So when you show you're working out here, you would do the 2 steps. So this would be step 1, and this would be step 2. You wouldn't have to draw the triangles, but what you could do is write 50,000 times 2 divided by 500, which gives you 200 micrometers. For our next section, it says most mitochondria are about 3 micrometers in length. The plant cell contains mitochondria, but the bacterial cell does not contain mitochondria. To use your answer in part A1 and information in the diagram to suggest why. This is worth 1 mark. If we look at the answer in part A1, it shows us that the plant cell is 200 micrometers and our bacterial cell is 2 micrometers. So straight away we can see that the bacterial cell is much smaller than the plant cell. If we continue looking at the information in the diagram, we can then see that the bacterial cell had to be magnified a lot more than a plant cell had to be magnified. So again, that suggests that the bacterial cell is smaller. Our answer here is the bacterial cell is much smaller than the plant cell. Question 2. The figure below shows a scale drawing of one type of cell in blood. Use the scale to determine the width of the cell, and you have to give your answer to the nearest micrometers. Look closely, we can see the diagram is represented on a grid. The grid also has a scale. The scale starts at this point and ends at this point, and that is equivalent to 2 micrometers. Using this information, we must find the width of the cell. The width of the cell is represented by these 2 marker points. If we replicate the scale, we can see from this point to this point gives us the same scale as the one below. So we have 1 unit here, we have another unit here, another unit here, and another unit here. Each unit represents 2 micrometers, and we have 4 of those units, which makes 8. So the width of our cell is 8 micrometers, and this is worth 1 mark. Linking words for today are magnification and resolution. Whenever we think of magnification, we should think of a bigger image, and when we think of resolution, we should think of a sharper image. Thank you for watching. Join me for the next video. Don't forget to like, share and subscribe to my channel.